I am the chair of the Medical Scientific Advisory Board, and I'm a father of a child with DYRK1A syndrome. So my son Ethan was diagnosed in the October of 2020, sort of during the pandemic. And both my wife and I were in research and clinical research. I was a professor at Duke, and I worked in oncology and advanced liver disease. And my wife was coincidentally at a CRO doing rare disease work. So she was already kind of plugged into the space. And so when Ethan was diagnosed, it seemed sort of like happenstance to have two parents that were involved in clinical research, especially one that was focused in rare disease. So we feel like Ethan was really a gift to us. And we kind of owe it to him also to do what we can with our backgrounds to advance the research in DYRK1A syndrome. So a little bit about myself there, and I'm happy to talk with anybody about that process because it was sort of interesting having two parents that were already in the space. We both have molecular genetics backgrounds, and we were very reluctant to even think that he had a genetic syndrome at the time. So I'm happy to tell that story at another time. But just wanted to get started here and talk a little bit about the Medical Scientific Advisory Board. These are our members. So you all know Amy, and then Dr. Breji Von is a clinical geneticist and has really one of the sort of only centers for DYRK1A patient care that's outside the US. And then we've got our own Craig McIntosh here in-house, and Lucas Bronicki, and then Dr. Amelie Pitton, Nadia, myself, and then I mentioned my wife Sarah. So she works at a foundation called NLORM, which is involved in developing genetic medicines for nanorare diseases. So that's a little bit about us, and I promise to do a better job next year, but before this family meetup yesterday, we had a Medical Scientific Advisory Board roundtable, and as I mentioned, we'll probably, I'll do a better job next year. But we had a few families attend, and really the purpose of that is to allow researchers who are working on DYRK1A syndrome to really present some of their latest findings and disseminate that to the wider audience, and then have an opportunity for us as Medical Advisory Board, and then you all as part of the community to ask questions, maybe things that you're interested in understanding more about DYRK1A and DYRK1A syndrome. And so yesterday we had that scientific roundtable. We'll do the same thing next year at the Dallas meeting. So probably on, I don't know, is it a Friday, Saturday, Sunday kind of thing, but Friday, we'll do another scientific roundtable, and I'll be sure to maybe get some questions ahead of time that you'd like to ask researchers, and then we can have them sort of contextualize that more into their talks and make it more applicable to areas that you're interested in knowing more about. So we'll work on that for next year. But just to summarize the talks yesterday, so the first talk was given by Dr. Martin Terra, and he's at the Jackson Labs. He was actually in Germany at a conference, and he was gracious enough to stay up late and provide his presentation. But he really has an interesting model system that he was working on. So, one of the challenges that you have in scientific research, especially when you're working with something like an animal model, which is usually a requirement that you have to, better understand complex diseases. But a lot of times in these model systems, they're just singular genetic backgrounds. And what that kind of means is it's almost like you're just studying one person versus studying a group of people. And in scientific research, that can kind of create problems where, maybe you test a hypothesis and you get a result, but maybe it's only applicable in this sort of one particular situation. So he really created, and his team created this sort of genetic diverse model system by which to study DYRK1A syndrome. And he had some really interesting findings in terms of sort of the almost clinical presentation in an animal situation of DYRK1A syndrome, and it was related to a pathway called BDNF, and you don't need to know what that is, but what it provides is really a good model system to understand more of like a population level, like all of us, all of our children and family members that have DYRK1A syndrome. It provides a good platform to study, drugs and pathways and things like that that can certainly help advance research in the future. So he was the first to present, and then Dr. Helen Wilsley, she's at UCSF, and she actually studies frogs and tadpoles. And her data was very interesting because, she had presented to our group a number of years ago, and what she heard from many of us is that, our children deal with a lot of constipation issues and GI issues. And using this, her frog and tadpole system, she was able to essentially show that using something like an SSRI, like Lexapro, which is kind of like an antidepressant, was able to help some of the GI issues. And that was related to DYRK1A kind of impacts these cells during development. There's these neuron cells that migrate into the gut, and these specific cells that end up in the gut are receptive to SSRIs like Lexapro, and so she kind of showed that that helped some of the gut issues. So that was some very interesting work that she had, and then Craig here also presented on some groundbreaking work that he's doing in the area of genetic medicine, where they're using these sort of small molecules that are similar to DNA. They're RNA molecules that are able to target specific mutations in DYRK1A syndrome. So he's working on one patient right now where he's using this approach to kind of correct for some of the consequences of the mutation of DYRK1A, which impacts the DYRK1A protein. So that was kind of the summary of what we had yesterday. And again, next year, we'll get more feedback from the group. And if you have any questions about the latest research or things that you're interested in better understanding with DYRK1A, we can help facilitate the discussion and speaker lineup so that it kind of helps provide more information to you all. So that was yesterday. So our objectives that we have just on a higher level for the MSAB are, first, a deepened understanding of individuals affected by DYRK1A syndrome. A lot of this is related to the fact that many of us have unique mutations, or our children that have DYRK1A syndrome have unique mutations. There's mutations that are in the gene, but we really don't have a good understanding of how that correlates to like the clinical presentation and whether there are other factors that are involved in that. So, one of the key pieces for us is trying to really deepen the understanding of DYRK1A syndrome, not just from that standpoint, but especially from a clinical standpoint. The second is to establish the drug development toolkit and a path to therapeutics. Being in the rare disease that we are, we're certainly growing in numbers. But the way that we would approach a clinical trial or understanding aspects of DYRK1A syndrome isn't the same way that you would do in like a general population. Many of those studies, you've got tons of people, and you can run what they call placebo-controlled trials, where there's a group that doesn't get something, a group that does, and you kind of compare. But we have such small numbers that we need to be thinking creatively and more proactively about how we can study DYRK1A syndrome, because when you have so few individuals and you want to understand whether there's a difference, you have to really understand a lot of aspects of the syndrome itself, both from how it presents, how individuals are different, what factors may influence that. And Kellen's here, and she's from Combined Brain, and she'll talk a little bit more about how work that they've done can help support a lot of these objectives. The third is establish a research funding mechanism. So we did do this year, and it was absolutely wonderful, and we'll talk a little bit about that in a minute. But we created a research fund to help entice researchers to look at many aspects of DYRK1A syndrome. So a lot of the gaps that we have in scientific research, we developed what we call an RFP, so a request for proposals, and we invited anybody that's done research really in DYRK1A to apply, and this year we're going to fund something. So we are. And so that's very exciting. So that's really going to catapult us forward in terms of better understanding DYRK1A research. We can allow other researchers in the space to receive funding to do so. The fourth is to define centers of excellence. This sometimes can be challenging, but really having an area or a place where there's clinicians that are very well versed in DYRK1A syndrome. So our goal is to establish centers of excellence, across the U.S. and certainly beyond, where they're going to have the expertise in a number of different disciplines under one roof. So you're not kind of bouncing around on trying to find clinicians across the U.S. or ex-U.S. that have expertise in GI or have expertise in clinical presentation or genetics. So that's one important area that we're trying to support. And then taking all that, to educate and communicate the science, the research, all these aspects of the MSAB to the broader communities like us, to really have you all be part of that understanding and partnership where we can, advance our knowledge base and research in DYRK1A syndrome. I just put this in here as well. So what we did this past year is what happens in science a lot is there's a cycle where you apply for research funding, you get research funding, you do an experiment, you do a study, you get your results. When you get your results, you publish them, you present them, you send them out to the scientific community. It gets peer reviewed, so other researchers evaluate the quality of the research and things like that. And so what we did this year in the journal Frontiers in Neuroscience is we created a special issue on DYRK1A syndrome. So it's open right now. It's open, I think, through the end of August. But much in the same way with the RFP, the funding mechanism that we created, we sort of outlined a lot of the gaps in DYRK1A research, just even the clinical understanding of DYRK1A, and we invited researchers all across the entire world to submit papers for that journal. So we already have, I think, several that are kind of pending, and one that it was finalized and submitted. So I just wanted to make you all aware that we're building it not just from a funding mechanism, but also from getting the knowledge out to the broader community in terms of peer-reviewed scientific journals. And so I did mention that we developed an RFP in our first funding mechanism for DYRK1A syndrome, and really want to applaud Craig here. So Craig applied for it, and he received our first grant for this funding mechanism for his work on the therapeutic that I mentioned that targets a specific patient with DYRK1A syndrome. So I want to provide a huge round of applause, if you will, to Craig. And, you know, I really just want to say that there's a lot of times that there are researchers that are, you know, brilliant in their own right, you know, and they do great work. But, you know, what Craig brings with him really is the passion for DYRK1A, the passion for our community. You know, I met Craig years ago, and, you know, you could just tell that he had the passion to do this. This isn't like, yeah, we were doing research and we came across DYRK1A and it was interesting and we did it, you know, and sometimes that's what happens in scientific research. You kind of follow the science, and maybe somebody was studying, you know, an autism gene and it led to DYRK1A syndrome and they focused on that. But he really led, I think, with the patients first. He saw the unmet need, and he has that kind of commitment, which I think is unique among, you know, researchers broadly, you know, not just even within rare diseases. So I just kind of wanted to, you know, say that about Craig, because I think he's, you know, just so well deserving of this award. So thanks, Craig. And I think, you know, yep. So we wanted to present Craig with this award.
Thank you very much.
For the grant. So it's $35,000 that you all helped support. So, you know, big congratulations to you all and to support this effort.
Thank you. Thank you, everyone.
That's so pretty.
Well, I'll put you on the spot if you want to say anything.
No, just like, yeah, because this comes from the community, I'd just like to say a special thanks to everyone in the room. As Oliver said, it is a passion of mine. I got into research to help people, and fortuitously, I think, through meeting Oliver and his wife, I've ended up in a place here in the US at a company that I think values all of those things. They put the patients first. So hopefully everything that we're trying to do is successful, and then we can update you next year on a bit of a success story in terms of a therapeutic towards DYRK1A syndrome. And yeah, I'd just like to express the gratitude from the NLRM Foundation as well. We're a non-for-profit trying to develop rare therapies for people who typically would not get a therapy due to the rareness of the mutation, and we provide that drug free for life. So this donation goes a long way to helping. Although it might be only one patient, in my talk yesterday, I highlighted how it could potentially be applicable to the broader community. So thank you, everyone. I really appreciate it.